These Frac, we call them Frac Group 7. So when we talk about Frac groups, Fungicide Resistance Action Group, this is a committee, international committee that classifies fungicides, keeps track of resistance development. And there's some new fungicides in the Frac 7 category. Alan mentioned some of those names, like Kenja, and so on. And so what I've been doing is testing these fungicides against the claims by the manufacturers were that they can control boscalid resistant strains. So fungicides in the same group, and this is unusual because usually you think about Frac groups, any member of that Frac group, you develop resistance to it. You cannot substitute any member of that same Frac group because they're going to be cross-resistant to all of them. This seems to be an exception. It's very interesting. I think it's good news because it looks like these fungicides are going to be useful for botrytis gray mold control here in this area. So this work involves a lot of people. Dave Dutton was a technician in the lab who did a lot of the assays. So this is a result. We've been screening for resistance, Alan mentioned, for about 3 years now. So this is a continuation of this fungicide screening, resistance screening effort that we've been doing. Delphi Hardeveld is a former postdoc. She was participating in this. Ola Kojar, PhD student, who you heard yesterday, she participated in this research as well. And I'll show you some of the data that she's been generating for this project. I've been receiving samples from Siva Savaratnam, BC Ministry of Ag. So we've got a pretty good picture of resistance in botrytis throughout this whole region now, British Columbia, Washington, Oregon as well. Some of the funding we received, so the Red Raspberry Commission, Blueberry Commission, the Pesticide Commission for Pesticide Registration, we had a block grant from the state, which supported some of this work, and also the BC Blueberry Council supported some of this work. So the disease cycle, just to remind you about the disease, it's a very good colonizer of senescent plant material, so it can colonize canes, it can infect green tissue, senescent tissue. We assume that there's a lot of inoculum out there in raspberry fields, berry fields all the time. This is the spore producing apparatus, so produces just one spore type, a conidium. They're wind dispersed. Olga yesterday talked about this infection process. We thought originally that all or most of the infections occurred through fruit or through flowers. The pathogen would infect flowers, sit dormant in those fruit, and then emerge under appropriate conditions later on. And so producing these sort of symptoms that you see here, this is where the name of the disease comes from, gray mold, this gray fuzzy growth on the surface of fruit, infects lots of different fruit. Widespread across a worldwide problem in fruit production, lots of different fruit crops. We have some questions now about how this process occurs, but the end result is this sort of symptom that you see later in the season. This can produce more conidial inoculum and reinfect fruit and so on. So it's a pretty simple life cycle. One spore type's produced. Very destructive disease under appropriate environmental conditions. Okay, so the fungicides that we've been testing, this is the list of fungicides we've been focusing on. These are the ones that have been used mainly for gray mold control in this area. So Alan talked about some of these. So Elevate was fenhexamide. I've listed the mode of action here. Also the Frac group. So I told you that this is Fungicide Resistant Action Committee. They've grouped these into they have different Frac numbers, which refers to the chemistry and the mode of action. Okay, so you can see the groups here. Rovrel or iprodione. Switch is a mixture of two fungicides, saprodinil and fludeoxonil. So you can see the two components here. The two components are in different Frac groups. They have different modes of action. Pristine, Alan talked a little bit about Pristine, has a boscalid component, also a pyraclostrobin component. The pyraclostrobin component has limited efficacy against gray mold, so we focused mostly on the boscalid component of Pristine. So those are the fungicides we focused our screening efforts against. The assay that we use mostly is pretty straightforward, so you can take strains of botrytis. So here it shows four individual strains. You can put them on a Petri dish. This is a Petri dish without fungicide incorporated into it. Okay, so you can see that after 20, this is after about 2 days of growth, 48 hours of growth. They grow on plate. We can measure that. This is the same 4 strains in the same orientation. So this strain is the same as this here and so forth. So this is a plate where we've incorporated the fungicide. And we do some preliminary testing to figure out what that rate should be. And you can see that then the response to this fungicide, when you take these individual isolates and put them on the fungicide-containing agar, so for example, this strain grows a certain amount here, but when you put it in the presence of the fungicide, it's greatly inhibited, okay? Some of these different responses, so this strain you can see here is inhibited slightly, so they're a little bit less growth here compared to over here. These 2 strains really aren't inhibited much in the presence of the fungicide, you can see here. Yep. No, yeah, that's right. So it's incorporated in the, so that we're looking at a top view of the plate. And so this has about, half an inch of agar medium in it, solid, kind of like jello-like stuff. And the fungicide is incorporated in that at the same concentration throughout the whole thing. So it's throughout this whole plate. Yeah. No, well, it's throughout the whole plate, but this is the only strain that's really strongly inhibited. So this one's much less, this strain here is much less sensitive to the other ones, okay? So these strains here, much less sensitive to the fungicide, and then we do some additional tests to ask the question of whether we predict they be field resistant under field conditions, okay? So this is some data for iprodione. And so the kind of data you get is, this is the individual isolates here. And on the y-axis, we have proportion growth of control. So we take the percentage growth that we have in the presence of the fungicide, divide that by the growth without the fungicide, and then we get an estimate of how sensitive it is. So you can see quite a bit of variation here. So we have one isolate that grows about 70% of the control. So it looks like some of those strains that were not very inhibited very much. Quite a few that were sort of in this range from about 50% down to 20% or so, okay? And then a bunch down here that all the way down to basically completely inhibited, so very sensitive to the fungicide. So this is result from blueberry in Washington that we sampled in 2016, but fairly typical sort of response that we get. So this is all Petri dish data, right? So it grows either 0 or 20% or 50% or 70%. So then the question is, which of these strains, if any, are resistant to this fungicide, okay? So if we're spraying field rates of these fungicides onto raspberry plants or blueberry plants, which of these do we predict are resistant, okay? So are any of these resistant to field rates of the fungicide? So what we've done to answer that question is to treat raspberries. We had a raspberry assay that we've developed. We treat raspberries with field rates of the fungicide. Then we inoculate with different strains that have different sensitivity types and ask the question, can those fungicides at field rates, rates that growers would apply them, protect against those strains? And which strains? Which strains can they protect against? Which can they not? Any predictions from this graph? Which ones are resistant? Which ones are sensitive? This guy? Yeah. What about all these? Okay. Well, it's a little surprising, and I was just talking to Alan Schreiber about this. Initially, we thought that the bulk of these were sensitive based on some information from that we were basically, this sort of through here would be the cutoff that this might be considered resistant, but all these would be considered sensitive. But when we did a fruit assay that I talked about, we also have this other assay where rather than incorporate the fungicide at a specific concentration through the medium, we just take field rates. We estimate what a field rate of a fungicide should be, treat the surface of the plate, dry it, pour it off, dry it, So the surface of that entire plate is covered with field rate of fungicide. So we think that that might mimic how much would be on the surface of a plant material. Okay, and then what we do, we take different strains and we streak them, the conidia, the spores, onto these plates. And then 24, 48 hours later, see if the spores can germinate and if they can grow. And it turns out that this assay is really quite highly correlated to our fruit assay. So when we treat fruit, we get a very similar response to treating the Petri dishes with field rates of fungicide, streaking those spores and see if they germinate and grow. Okay? So this is then, so we've got field rates of Iprodon across this plate. We've taken 4 strains and we've divided the plate up into 4 sectors. So we streak conidia here and so forth. Okay. So we've got 2 cases where the spores have germinated and grown just fine. Another couple of cases where they're completely inhibited. Then we can relate that. So we select the ones based on the previous Petri dish assay. Here's the percentage growth of the control on 3 parts per million Iprodon. Okay. So here's some strains that didn't grow at all, so they're completely inhibited. Here's a strain that grew 10%. Here's a strain that grew 12%. Here's a strain that grew 20%. So this allows us then to create a cutoff for what we would predict for resistance. And so any strain, it looks like, that grows greater than about 10%, so 12% or greater, on 3 parts per million Iprodon would predict to be resistant under field conditions. So we've done that now for all these fungicides. So we can relate all of the assays that we've done in the lab with defined rates of the fungicide, calculate sensitivity, make predictions about whether we would predict under field conditions whether these would be resistant. Okay. Yep. You know, the microphone. Take the microphone. Otherwise, I'd have to repeat your question.
All right. On your Petri dish, not that I'm going to get into it, but anyway, do you take, like, say, a blueberry and slice off on a slim slice of it that's, is contaminated, or do you just scrape off the outside of the moldy part of the berry to get your sample to work with your fungicide?
Oh, okay. So the strains. So we usually wait until we see infected berries. So we see gray mold symptoms, and then we can pick those off. And the way that Siva was sending me samples, which works very nicely, is to take a cotton swab and just touch it to the surface of that sporulating fruit, and then he can just mail those to me. Under the appropriate permits, APHIS permits, of course, because they're coming from Canada. But he can mail those to me, and I can streak them out and culture them. Absolutely. Yep. So it's very, very neat method, actually. Okay, so I just want to share a little bit of data with you before we move on to talking about the Frac 7s, just to emphasize that Alan mentioned the problems with resistance that we have. So what we're finding is we've got high levels of resistance in a lot of fields. There's a lot of field-to-field variation, but we've got a lot of problems with resistance to most of these fungicides. One of the exceptions so far has been fludioxonil component of switch. Okay, and we found widespread saprodinil resistance. That's the one component of switch. To date, we haven't found evidence for resistance to the fludioxonil component, and no one, or it's very rare anywhere in the world that someone has found resistance to fludioxonil. So what I did was I wanted to, and so what our assumption is then is that switch is still active against Botrytis cinerea, even if you have saprodinil resistance in your field, because of the other component, because of the fludioxonil component. So it's basically functioning as a standalone chemical if the saprodinil component is gone, okay? So what I did is a little experiment with those swabs, so using the streaking method, okay? And what I did was to streak, I selected a number of isolates, and what I did was select isolates that were mostly based on saprodinil resistance. So the ones that we knew were saprodinil, I took about 50%, half or so that were saprodinil resistant, so that's the one component of the switch, another 50% that were sensitive to saprodinil, and then tested them by streaking spores on field rates of these chemicals, okay? So separately, so I took switch, I streaked them on plates with switch, so that's the mixture. I streaked them on plates just with the saprodinil and on just with fludioxonil. So Alan mentioned that fludioxonil alone is not registered on berries, but it's registered in other crops as a standalone product, so I had access to it. So what you can see, okay, is I selected, as I said, I selected saprodinil resistant and sensitive strains. So you can see down here, the data are zeros or hundreds, and that's just percent germination of conidia. Okay, so I streak the conidia on those plates, and then go back 24 hours later and say, have they germinated or not? All right. So in the presence of field rates of saprodinil, you can look down here and see that in some cases we have 100% germination. So that's a saprodinil resistant isolate, zero germination, saprodinil sensitive isolate, all the way down here. Okay, so we have about half or so, probably a little more resistant ones that are germinating and growing just fine. The others not. Okay, look over here. Same. So these are all the strains, and I sampled these from different regions. This area. Look at switch. So switch is controlling, is doing quite well. So no germination for some of these strains that are saprodinil resistant. So in these cases, then switch is still effective, even though you have saprodinil resistance, okay? Look down here, same thing with the fludioxonil component, so there's no germination there, all right? But look at these. This one here, one strain, 100% germination, grows just fine in the presence of switch, saprodinil, fludioxonil. Another strain here, same thing, okay? I can see Randy grimace. Another one here that's interesting, it germinated in the presence of switch, but not with fludioxonil by itself. So what this suggests to me is that we do have fludioxonil resistance, and it seems rare right now. We need to follow this up and check out some more strains. But what it does is emphasize to me that the situation is very dire. If there really is fludioxonil resistance and that increases in frequency, switch is gone too, okay? And that was one of the few remaining chemicals that you had. All right, so that's just to emphasize the situation we're in. This just shows, we're gonna be talking about Frac 7 fungicides, which is in our same group as boscalid, okay? So this is boscalid resistance in raspberry in 2016. Data is presented a little different way, so we do the assay on the agar media, we do the fruit protection assay, we do the spore germination assay, and we determine which of those strains are resistant. We predict are resistant. This is the proportion of isolates that are resistant in the hatched bars, hatched portion of the bars, okay? So they adds up to 100% or 1. The solid color portion of the bars are the sensitive strains, okay? So they add up to 100. This is Washington raspberry. So on average, we have about 60% resistance. That's about what we found over 3 years of sampling, about 60 to 70% boscalid resistance throughout this region, okay? BC, Washington, Oregon. So here's for British Columbia. It's about the same, a little bit higher for this sample. It really depends on, it varies a lot sample to sample. The fields, so each of these individual bars represents a field, okay? And what we found is that there's a lot of variation field to field. So you can see that here. So for example, this field, there's a high proportion of sensitive isolates to boscalid, small proportion of resistant. This field over here, exactly the opposite. So mostly resistant strains, okay? So that 60% or 70% average is a bit misleading because you have a lot of variation field to field, all right? And that variation field to field we found is a result of fungicide use patterns in those fields. No surprise there. So low input fields organic fields, we have generally low frequencies of resistance. More intensive use of fungicides, we have higher rates. Okay, but lots of variation field to field. So when we say we got a lot of resistance problems, some fields don't have the problem. Okay, so that those fungicides can still, certain fungicides can still be used. So boscolids can be still effective in some fields, like say this one, you can still use boscolid. This field, probably not. Okay, so we've got a real problem with resistance. So this is the fungicide list that we've tested. These are the fungicides that we know we have resistance problems. Okay, almost all of them. I said we had up until recently, we didn't have any evidence that we had fludioxonil resistance. I have a question about that as well. Yes, it did. Not far from here. That's correct. Blueberries, I think. Okay, so that's a good segue then into the Frac 7 fungicides. So Alan talked a little bit about these. He's been doing efficacy testing with these. They look really promising. So they have the same mode of action. So they inhibit the same enzyme in the mitochondria. It's basically part of the energy generating machinery of the cells. You have them in your cells. Fungi, we're very closely related to fungi actually. So it's the same system. Our enzyme's not sensitive to these, but it affects the energy metabolism of the fungi. I said we had widespread resistance, so about 70% on average. So some of these new products, so Alan mentioned some of these, Kenja, okay? Isofetamide is the active, adepidin from Syngenta, flopiram, penthylpyride, Alan talked about Fontellus, fluxapyroxad, okay? So there's several of these products that they're recently developed. Some of these are registered, some are not. The claim by the manufacturer is that these can control boscolid resistant strains. So even though they're in the same Frac group, the claim is they can control boscolid resistant strains. Okay, this is really unusual because you don't expect this within a Frac group. You usually say every member of a Frac group, you cannot substitute one for the other. The good thing about this, the boscolid resistance, is we know what the mechanism is. Okay, so we know at the molecular level, we know what the mechanism is. We know what the specific mutations are that confer boscolid resistance. So the efficacy of these new fungicides then depends on what boscolid resistant mutations you have in your area. Okay, so that's what we've been focusing on. So this is, when you think about cross resistance, that's why the Frac group classification is really important. So if you have decreasing or increased resistance to, say, fungicide A, increasing resistance to fungicide B, so you have some sensitive population down here, and you have some less sensitive or resistant population up here, this is classical cross resistance, okay? So that within a Frac group, you generally see this sort of pattern. So if you're resistant to one member of a Frac group, you're going to be resistant to everything else, every other chemical in that Frac group. So this is the general pattern. So these new Frac 7 group fungicides it's an interesting phenomenon because it doesn't follow this pattern. Okay. So even though they have the same mode of action, it's slightly different. The mechanisms of resistance are slightly different, and there's potential for these to still control resistant strains to other members of that Frac group. Okay, so what I did first then was to take a sample of boscolid resistant strains from throughout this area. So I had a sample that were boscolid sensitive, boscolid resistant, and this is the sort of pattern that we see. So this is what we call EC50 value. It's just a little more sophisticated approach to estimating the sensitivity. Use more concentrations. So it varies a lot. So these boscolid, the highly sensitive strains are down here in this range. So 1 part per million, 3 parts per million, and so on. But there's a wide range of sensitivity up to into the 500 parts per million range. So this EC50 means estimated concentration to inhibit 50% of growth. So it's just a way of estimating how sensitive something, a fungus is to a fungicide. So we have this wide variation, okay? Through our fruit assay and our spore germination assay, we know that the cutoff for boscalid resistance, field relevant resistance, is about here, so about 8 to 10 parts per million. So these are sensitive strains here. These are resistant strains here. But what you can see is there's a wide range of within this resistance group, there's a wide range, okay, from 20 parts per million to 540, huge range. So what we did then was, we have these strains that are sensitive and resistant to boscalid. Then we asked, what's the distribution look like for those same strains to these new Frac 7 fungicides? So those are listed here, okay? Adepidin and so forth. Okay, there's adepidin. So all of these boscalid sensitive and resistant strains, they all had EC50s of about 1 to 3 parts per million, okay, down in this range. Isofetamide, same thing, all down in this range here. Flopyram, a little bit wider distribution. Most of them are down in this range, but a few up in here around the cutoff for resistance for boscalid. Okay. So a little bit more variation. Penthiopyrad, very similar, mostly down here, but a few up in this range. And actually this one here is really interesting. It's quite insensitive. Okay. And then fluxapyroxide, same thing, and another strain up in here. So what this means is that it looks like most of these fungicides can control the boscalid resistant strains that we have here, okay? But there's a few exceptions. So a few of these isolates in this range, and here to some of these chemicals, look like those are probably going to be resistant strains. We haven't completed all the fruit assays and the spore germination assays to say for sure yet. We're working on that. But it looks like there might be resistance out there already to some of these, but in general, they appear to control most of these boscalid strains quite well. So that's really encouraging. Okay. So then you can ask, well, what's the relationship when you compare sensitivity to boscalid and to these other chemicals, right? So you take the sample of isolates and you select in a certain concentration, discriminatory dose for each of these chemicals. So what I've got listed here are the response to boscalid, so on the x-axis in both cases. And so you can see the typical distribution here. These are the sensitive isolates down here. These are the resistant isolates up here to boscalid. Okay, so then you can ask, well, when you challenge those against flopyram, what do you see? Well, what you see is, and remember that classic cross resistance pattern was something like this, right? So you had sensitive here, resistant up here. So you're not seeing that pattern here. You're seeing a slightly negative relationship rather than this positive relationship. Same thing for isofetamide, okay? Same kind of response. Adepidin, penthiopyrad, fluxapyroxide, a little bit different. 1 minute. Ooh, I gotta move. A slightly positive relationship here, okay, for these three. So slightly different. So this suggests then we've got, among these new Frac 7 fungicides, we've got 2 functional groups, okay? So we've got a group, isofetamide and flopyram appear to behave similarly, and then these fungicides, adepidin, fluxapyroxide, penthiopyrad, appear to behave similarly to each other. So 2 different groups. So the other part of this then is we know the molecular mechanism, so we can we can sequence part of the gene that's the target of these fungicides, and we can ask what specific mutations are present. The mutations that we're interested in are here. So these sites are named like this. So the mutations, when you say H272R, means this particular location in the gene, substitution. This is the sensitive or the sensitive state here, sensitive to all of these fungicides. This 272R mutation confers resistance to boscalid, okay, but not to the not to the others. 272Y to boscalid to these two fungicides, and then but not to these. And V resistant to everything. So I really only have a minute left. So I must have spent way too much time at the beginning. He's going to donate some time. Thank you, Alan, because it's, I got distracted there early. I'm sorry about that. So what we did initially then was to select 22 isolates. We had 11 sensitive to boscalid, 11 resistant, and Olga characterized the mutations. So this is what the site looks like. So what we found was 45% of them had the 272Y mutation, 46% had the 272R, and 9% had the 272V mutation. Okay. She did a larger sample fairly recently, so it was 104. Selected some isolates from different crops, boscalid resistance, and then this is the frequencies of those different mutations here. She still hasn't been able to determine if it's not one of these two, what else it is. So there's this other category. But what you can see is a larger sample. We have about 65%, the H272Y, about 30% of H272R, and then about 11% that's other. Okay, so that's where I think that we can make a prediction that in general, at least some of these new Frac 7 fungicides are going to perform pretty well for us because of the mutations that we have. The concern is these others. Okay, if they're those V mutations, those are conferring resistance to everything. So if those turn out to be in higher frequency in certain fields, they're not going to work. All right. So we want to characterize these mutations on a larger scale and see how much variation there is field to field. So in summary, then, so these new Frac 7s look promising. But want to emphasize there's evidence that we might have resistance already to them. Okay, so it's existing, it's out there. If they're overused, we're going to have exactly the same situation that we're in now with all these other fungicides, the ones that have been used previously. So we have to use them very carefully. Appears to be two functional groups, so I think we can consider flopyram and isophetamide as similar in terms of the resistances. Also, and then the other group. It looks like we've got these 3 mutations in this area. Okay, so in general, the 2 of them looks like they're sensitive to most of these Frac 7 fungicides, but the V mutation is of concern. What else? Yeah, so this V mutation, that's the one we need to watch out for. We want to determine what the actual frequency is of that among those other categories, do a larger sample, see what frequency it's in, how it's distributed around. But in general, these fungicides, I think, look promising for this area for gray mold control. So, okay, so I'll finish and take any questions, and thanks for the extension. Nobody has any questions?
Mm-hmm. Like say some of the people that's got their field down in a valley where they like the fog or kind of lays down low, rather than kind of being on a little bit of a hillside where you get air dumping and at least amount of the problem. One other thing I recognize, and it hasn't been spoken on very much, is a lot of times you'll look at blueberries and you'll see like botrytis, the flowers rotting, but a lot of times a spanner worm will cause the same problem that thinks you've got a mold problem.
Oh, okay. So symptoms that look similar.
Yeah, they kind of eat it off at the stem.
Yep. Any other questions?
So Tobin, in your documentation of fludioxonil resistance, was in those samples, was the spray just fludioxonil? Was there any combination of any other?
That's a good question. I don't know. I don't have the spray records for those fields, so I don't know. But it wouldn't be. It's not registered, so I assume it's being treated with switch probably.
Would there be any like Cap 10 a tank partner with it? Do you know?
I don't know. Do you know, Steven? You know if there's Captain was being used? I don't know.
Because that's the situation we came when Elevate had its problem, when we've had problem with Pristine, and when we've had problem with some of the other fungicides. When we went on this Captan free program in 2010 and 11,
Yeah.
it was 12 that just slammed us with resistance.
Yeah. Yeah. Yeah, it could be. You know, it's that's why we recommended Captan as a companion for all the, you know, for every spray that goes on, because it's a good strategy, because it's a broad spectrum, and that's a really solid resistance management strategy to mix that fungicide with a site-specific one. So, yeah. Yep. Chris.
Remote question. Comment question. A lot of this resistance work is also being done in grapes. They are noticing that plantings near orchards have more resistance. Could there be an interaction like this for blues in diverse landscapes?
Near porches?
Orchards.
Near
Sorry.
orchards. Yeah, well, it's, you know, it's an airborne fungus. You know, these spores can move through the air, so there's potential that resistance selected in one location could be moved over. So even though someone's not using those particular fungicide, you can still detect resistance in them. We have some evidence for that, but by and large, what we've been surprised by is that even fields that are quite close together can often have really different resistance profiles. That's what we're finding. So we can't make predictions kind of regionally about, you know, everybody doesn't have boscalid resistance. We can't make that prediction kind of on a regional basis because we have such variation field to field. So we need a way to study fields in more detail, each individual field.
Thank you, Tobin. We've run out of time. Our next speaker